Hello everybody, I hope this video finds you well, safe, and healthy. We're going to use this time together to talk essentially about dynamic instability, about microtubule formation. Talk a little bit about the why of dynamic instability at the very end of this video, but we'll focus on the mechanism for now. So first thing worth noting is that all microtubules originate from the centrosome. The centrosome is a large spherical structure inside the cell, in the cytoplasm. And the centrosome is studded with rings of a protein called gamma tubulin. Now, I'm going to have to draw everything in 2 dimensions, obviously, because that's what the screen I'm working on is, in 2 dimensions. But I'd like to point out that really everything we're talking about today is going to be rings, hollow rings or cylinders. So this structure that I've drawn here on the surface of the centrosome is actually a ring of gamma tubulin, such as this. So if this is the ring kind of looking out of this, so I've got my hand in front of me, that's how I'm visualizing this. If I'm looking at my palm, I'm looking at the surface of the ring of gamma tubulin. Then when we see it here on the edge of the centrosome, it's like taking our hand and rotating it. So we're just looking down at the profile of our hand, which makes it that much thinner. Such is the same for the microtubules themselves, because on these gamma rings, on these gamma tubulin subunits, we're going to be adding alpha-beta heterodimers of tubulin. So this is an alpha-tubulin protein, a beta-tubulin protein that have come together into a heterodimer, and these are going to be added onto the gamma ring and then added onto one another to form a microtubule. Now, to take a little peek into where we're going with this, that microtubule in profile is going to look something like this. Each one of these chains of tubulin, each one of these individual chains of tubulin is a protofilament. But again, just as we said for gamma tubulin, imagine if we take this microtubule here and we pull it out of the screen and we rotate it 90 degrees, so now that we're looking down the barrel of it, that would look like a hollow cylinder or tube. I don't know if I have 13 drawn here, I wasn't counting, but each microtubule is made up of 13 protofilaments put together into one of these cylinders or one of these tubes. So that's where we're ultimately heading in this narrative. So we've got our heterodimers of alpha and beta tubulin. For those of you who are also taking the lab of this course, the TUB1 gene actually encodes alpha-tubulin. So this is the protein that we're trying to label with GFP right now, but that's besides the point. These tubulin dimers have a number of fairly interesting characteristics. One, they can bind to GTP. And when I fill these in, this is going to be the GTP bound form of these tubulin dimers. So right now, this alpha-beta heterodimer is holding on to GTP, guanosine triphosphate. But these tubulins are also GTP aces. They have their own intrinsic GTP ace activity, which means that they can cleave GTP from guanosine triphosphate into guanosine diphosphate, and then they release an inorganic phosphate as a byproduct of that reaction. So these tubulins can do that cleavage and convert GTP into GDP, or guanosine diphosphate. And when I draw these and I draw them open, that's going to be our GDP bound form. That way we can tell them apart while I give the explanation. Now, again, this cleavage of GTP to GDP is intrinsic by the tubulins itself, but the tubulins are pretty crappy at this. They don't really do a good job as a GTP ace, so this takes some time. There's a lag here, a time lag, in order for this cleavage to occur, which actually benefits us. And the reason why that benefits us is because any tubulin that is bound to GTP is stably formed in a microtubule, and any tubulin bound to GDP is unstable. It doesn't really like to be part of that microtubule, and it would rather be free. It would rather be a free dimer than part of the microtubule. So really quickly, let's review all of these ground rules. Alpha-beta heterodimers of tubulin can bind to GTP, and when they do, they readily and stably interact with microtubules, and they're happy to be there. Those tubulin dimers cleave that GTP on their own after some time to GDP, but any tubulin bound to GDP is not happy and is unstable in a microtubule. So let's see what's going to happen. So in the cell, of course, there's a lot of floating tubulin dimers around. There are a lot of them floating around. And some of them are bound to GDP, and some of them are bound to GTP. So some of them are bound to GTP, and some of them are still holding on to GDP. So we have a mix of these two different types of tubulin dimers in the cytoplasm. Now, of these, which are going to readily and stably interact with gamma tubulin and begin to form a true microtubule? Well, it's the tubulin dimers that are bound to GTP. They will readily bind here because that's what they like to do. They're stably found in these microtubules. That's where they like to be. Now we can give some more definitions because we're actually beginning to build a microtubule. We have the plus end of the microtubule is the growing end, the end that's pointing away from the centrosome, and the minus end is here. That's the end that is embedded in the centrosome, the end that's interacting with gamma tubulin. Additionally, the tubulin subunits that are at the plusmost end are the alpha subunits, and the beta subunits are interacting with the negative end. So this is alpha, beta, right to left. Okay. All right, so that's going to continue for some time more. And by that, I mean we're going to keep adding tubulin dimers here. And if we're adding stable tubulin dimers, then they were all bound to GTP. But time has passed, hasn't it? So from the time that we added the first few dimers to the time that we just added these, time has continued. And when time passes, what do tubulin monomers do? Well, when time passes, tubulin monomers cleave their GTP into GDP. And that's what's been happening here near the minus end of this growing microtubule. Now that's okay, because yes, while it's true that this portion of this growing microtubule is now unstable, because these are GDP-bound tubulin monomers, we have this stability here given by the dimers that are held onto GTP. So this microtubule, as it's forming, will not disintegrate. It will not fall apart because of the stability given by its cap. And I've just used that word. This is the GTP cap of this growing microtubule, and it will keep the microtubule stable as it continues to grow. So let's do that. Let's continue to grow this microtubule. So more time passes. Well, more time allows more tubulin dimers to be bound. Now these dimers do join one at a time. You see me, I'm actually drawing them two at a time just to save myself some time of drawing. But they join one at a time. They're all holding on to GTP, which is why I'm filling them in. Otherwise, they wouldn't be joining in the first place. But while all that was occurring, time was continuing to pass, and so the tubulin that was previously part of the cap is still there, didn't go anywhere, but it cleaved its GTP to GDP, and so now that tubulin is unstable, and the cap has moved forward. Still stably growing, still has a GTP cap. Now let's say that this microtubule actually reaches its target. Maybe the target here is the Golgi. Well, at the Golgi, there are proteins in the Golgi membrane, and those proteins are loosely, collectively, referred to as anchoring proteins. And these anchoring proteins do, just as I've drawn them right here, they grab onto the end of the microtubule when the microtubule makes contact with that target membrane, and they squeeze and hold that microtubule in place. Now, time has continued to pass, hasn't it? So if time has continued to pass, even these microtubule dimers, even these tubulin dimers at the GTP cap have cleaved their GTP, and they're now holding on to GDP, and essentially this entire microtubule is now unstable because all of the tubulin dimers are holding on to GDP. Well, too bad, microtubule, because the anchoring proteins are holding you in place. And so there's no opportunity for this microtubule to break apart, to fray, and to collapse because it's already been anchored. So is every single tubulin monomer that's part of this microtubule chemically unhappy being there? Yes. Is this microtubule stable in the sense that these dimers are preferentially staying in that microtubule? No. But the microtubule is anchored in place, and it will not fray or collapse in that way, in that manner, because of the anchoring proteins. Now, why do this? A number of you asked in Edpuzzle or in your whisks why this is done this way at all. What is the purpose of favoring disassembly? Well, let's stop for a moment. Is this cell in mitosis right now, or is this cell kind of doing its own thing, being its own cell in earlier stages of the cell cycle? Well, we know this cell is not in mitosis, because in mitosis, all of the microtubules are engaged in being spindle microtubules, spindle fibers. And they'll soon be pulling chromosomes to the metaphasic plate in metaphase, and then pulling sister chromatids apart in anaphase. That's what microtubules do in mitosis. This microtubule right now is a highway. It's a protein cable. It's here for the purpose of transport. So this cell is not in mitosis. This microtubule is doing its transport thing, its regular cell thing. But let's say this cell does enter mitosis. If the cell enters mitosis, then all of these tubulin dimers need to be repurposed. They all need to be released so that they can build mitotic spindles for the separation of sister chromatids. How can we possibly, rapidly, spontaneously, all at once, release all of the tubulin dimers that are present in the cell at any one time? Well, we can do it actually fairly easily. All we have to do is pop the anchoring proteins. If these anchoring proteins give way, essentially if they release the microtubule, well, we have to ask ourselves, all of these tubulin dimers are holding on to what? GDP. And if they're holding on to GDP, they're unstable. The only thing keeping this microtubule in place was the anchoring protein. And if the anchoring protein has given way, all of these tubulin dimers will fall apart. They will all go back to free tubulin dimers in the cytoplasm, and they'll ultimately give up their GDP, take up GTP, and be ready to form new microtubules. Except, if we're in mitosis, those new microtubules are actually going to be spindle microtubules set up on opposing poles of the cell for sister chromatid separation. When mitosis is done, all of those spindles are unneeded. That's not what you need microtubules to be at the end of mitosis. You need microtubules to go back to being protein cables for vesicle transport. And so all of those microtubules spontaneously disassemble again, and then they are repurposed and repackaged as microtubule cables for intracellular transport. So we have this very dynamic flux of microtubules where during mitosis they have to be spindles, and not in mitosis they need to be cables. And this dynamic instability, this inherent instability of tubulin dimers allows the cell to rapidly break microtubules down at a moment's notice so that they can dynamically go between these two different functions. Okay, so that's that. Let's tell one more side of this story. Let's get rid of this and kind of back up a few moments ago to the growing microtubule. And let's just give it its cap here. Well, as microtubules continue to grow, what are they incorporating into themselves? Tubulin dimers. And if more and more tubulin dimers are becoming part of growing microtubules, what does it make sense there's less of in the cytoplasm of the cell? There's less free tubulin dimers because more and more tubulin is already engaged in microtubule growth. So if this microtubule misses and it doesn't land on a target membrane in a rapid enough time, and if the levels of free tubulin in the cytoplasm continue to fall because more and more microtubules are being made, then the rate of growth of the GTP cap is going to slow. In other words, the GTP cap growth is going to take more time. And as time continues to pass, more and more tubulin on the back end is going to cleave its GTP into GDP. And if the pools of free tubulin get so low and the rate of new growth slows down so much that even these tubulin dimers at the cap have enough time to cleave their GTP into GDP before new tubulin dimers have been added, well, now the entirety of this microtubule is unstable. All of the tubulin is bound by GDP, and there's no anchoring because we missed our target membrane. That means this entire microtubule is going to spontaneously collapse. All of these tubulin dimers will disassemble and go back to being free dimers in the cytoplasm. This entire microtubule will fray and collapse all the way back to the centrosome, possibly. And in the cytoplasm, once free, more and more of these tubulin dimers will exchange their GDP for GTP and once again be willing to grow, to join gamma, and to begin growing a new microtubule that has the potential to perhaps hit its target, and we can try once again. So the analogy I use in the video is this idea of casting out a fishing line. And every time you miss a target, anytime you miss a fish, you reel that all the way back into shore and you throw it out again. The only way that you can catch anything with a random approach, and that's catch a fish by randomly throwing a line into the water, or catch an organelle membrane by randomly throwing a microtubule into the cytoplasm, the only way you have any chance of success doing that is you have if you have this dynamic iterative approach where cast it out. If you hit, great. If you don't hit, reel it in and cast out again. And that's exactly what microtubules are doing. So that's dynamic instability. Take-home messages from dynamic instability is that tubulin dimers containing alpha and beta tubulin can bind two forms of GTP. GTP itself, which is the stable form, and then they cleave that to GDP, which is the unstable form. Stable microtubules, stable dimers, with GTP bound continue to grow on the growing end, the plus end of a microtubule, but in time they're also cleaving that GTP to GDP and becoming unstable. As long as we have a cap of GTP containing tubulin, or we've been anchored at a target membrane, growing microtubules will be stable. If we miss a target membrane and we can't be anchored, and enough time has passed that even the tubulin in the GTP cap has cleaved that GTP to be GDP, and all of the tubulin in that microtubule, in that unanchored microtubule, is GDP containing, then that microtubule will fray and collapse, releasing all of that tubulin dimer back into the cytoplasm so that it can try once again.